Okay, hello everybody. This is Psychs 3280 Neuroscience of Consciousness, week 5. And this week we are going to talk about eyes, IRE visual system, and its relation to consciousness. All right, so let's first start from the recap of the week 4. So this is the 3 key figures from the Stan Dehaene study in the 2001 in Nature Neuroscience. And the first, what we discussed was non-conscious processing and the sandwich masking. The word sandwich masking refers to the fact that this particular type of the visual masking they made was composed of both forward masking, that mask starts before the target, and then the backward masking. And backward masking is usually much more stronger, but you know, in their case, they combine both feed forward and backward masking to get the maximum effect. And in particular, so this is going to be as a direction of the time, it's going forward like this way. This or that is considered as a forward masking, and then this too that follows the target is a backward masking. And under this condition, what they found was that the behavior first validated the conscious visibility. Under the visible condition, they found that the subjects were able to report spontaneously and also name the word, and also they were able to remember afterwards and so on. And then the invisibility of the masked stimulus, so they could not spontaneously report and could not name and could not remember things. And the blank or distractors that serves as a false alarm rate estimator is not different from this masked situation. And finally, the fourth choice paradigm to say, let's say, after seeing this, after showing note, you ask subject to choose whether a note or a lion was present, and they were at chance. So this is a very strong, stringent criterion of the behavior invisibility. Okay. And then further on, they also established the neuronal and the behavior evidence of non-conscious visual processing. So here, neuronal evidence corresponds to this, activation of the fusiform or word form areas, which coincides with the visible world activation, here, the green. But a visible world activates both parietal and also frontal areas, which we'll talk about most likely next week. And that's a much more extended and stronger activation. And then the behavior evidence-wise, I showed the repetition priming effect. And under this kind of situation of impossible to discriminate, people still show the reaction time advantage for the repeated words. That indicates that there is some kind of processing going on. All right. And then next, at the next part, I also talked about the eyes and also its structure with relation to the retinal ganglion cells, cones, which is responsible for our color vision, and the rods that is responsible for our night vision and blind spot. And in particular, I talked about the blind sight behaviors in monkeys, and also which allows us to go into the more refined understanding of what neuronal mechanisms are underlying the blind sight behavior in monkeys. They deny seeing, but they can localize by detection task and also eye movements. And then I also introduced the metacognitive accuracy as a way to discriminate conscious from non-conscious processing. The idea is to consider hit and correct rejection trial in a signal detection task as a target present for the subject, and then miss and false alarm as incorrect trials for the subject. And then the subject's task is to discriminate this, correct from incorrect trials. And if you know when you are correct, and if you know when you are incorrect, then this metacognitive accuracy measure that considers high confidence followed by the correct rejection and hit as hit, second order hit, or meta level hit, and also a false alarm and a miss, which is a mistake, followed by low confidence as again, correct rejection, and false alarm and miss is also defined in a corresponding second order way. We can define and quantify how accurately people know about their own decision. And it's shown that in the blindsight subject, when subject claims that they cannot make an cannot see it consciously, then even though they have a strong motivation to earn more money, they can't utilize that information. And that is a very compelling evidence to show that subjects are in fact able to still behaviorally, objectively demonstrate the information processing that is not available for conscious awareness. So metacognitive accuracy is a way to discriminate conscious from non-conscious processing. Okay, and then the structure of the eye led to our discussion towards the end that do we see color outside of fovea? Just quickly reviewing. So here is an eccentricity 0 degree that is a fovea here, and then you are seeing something here, direct fixation. And then as you see the cone density, okay, it's a density, which is important, declines sharply, and then it's still above 0, but goes quite sharply declining. And under this situation, like 20 degree from the fovea, are we actually seeing the color? That's the question. And we are going to start there for this lecture. All right. So in week 5, we will explain the properties of visual cortex, in particular primary visual cortex, or V1, and then discuss the implication for our color vision or any other vision actually at the periphery. And also explain the neural mechanism of the blindsight. We don't know fully understand, we don't still fully understand blindsight, but to our best knowledge, I'll try to give an explanation. And while going through the Ariel visual systems and alternative pathways, and then there we encounter a couple of important questions as well as some concepts that will be followed up in the following weeks, starting from here. Okay, so the learning objectives of this week is as follows. First, we are going to ask again, what is our peripheral experience? Okay, what do we actually see in the outside of fovea? And for that, we are going to do a closer look with the color and the motion, and also using some kind of informal metacognitive measure to see what we are actually experiencing in the periphery. Okay, that's the first. And second, we are going to look at what neural mechanism underlie the transmission of visual inputs from the eyes to the brain. This is partly reviews back last week. And then what is a receptive field of a neuron? This is a very important concept. And then what are the key properties of the V1? That is primary visual cortex. Basically, it's a columnar organization and layer structure. And then what are the implications of these properties for V1 for conscious phenomenology? And also another property of the V1 is this cortical magnification and the retinopathy. And that leads to the hard problem of consciousness quickly. And then what are the visual pathways from the eyes to the brain and its implication for the blind side? I'll just give you a quick answer that there are multiple pathways, and some of them are most likely underlying blind side. And for this week, the amount of the chapter that is covered is relatively huge, but part of the last part of the chapter 3 and the 4 is going to the main parts. And then chapter 8 is also implied. And chapter 5 to 7, I also uploaded, but you don't need to read. It's an optional reading. For those people who wants to go the chapter from 1 to 8, you can read it through, okay? So let's recap the demonstration in week 4. If you haven't done this demonstration by yourself, please do it right now by stopping the video and then examine your experience. Okay. So first thing you need to do is to put your hand extended like this, and then adjust the distance from your display so that your thumbnail roughly coincide with the thumbnail that you're seeing on this display. Okay. So now you're supposed to put your finger thumbnail around this figure center and then examine what your peripheral experience is like. And for me, I can kind of see there are lots of colors, especially the bright ones, the yellow, but also I can see lots of missing horn-like thing. Maybe it's related to the background light conditions in the room. But also, I can't really see which color I'm seeing at which location. I have a vague impression of there are colors, but not really clear. Probably, if I'm asked, I may not be able to tell which color I'm seeing at which point. Okay, and then now, if you do the same thing, but locating the finger to the bottom and then see this figure. And if you're like me, I would say that I can actually comfortably see all of the colors, and I can also indicate the location of the color quite confidently in both left eye and the right eyes actually. Okay, but if you haven't completed, please participate in the survey for week 4, and then we will collect that information and then discuss in the later weeks. Okay, so this week I'll add a little bit more of this metacognition of your peripheral experience. So, what I bring is this trump card, as in this thing, and so if you have one of these, trump card, then please prepare right now. Okay, this is important to do it by yourself. Okay, so here is the direction. One is the preparing a set of the playing card suit, and then pick a card with the back side facing to you. So I'll do something like this, and then move the card to the place where you think you can identify the identity of the card, like both color and also the number of the card. Okay, so if I were to do that, then maybe I'll do. Now I can actually see the card identity, so it doesn't work. But I'll just hide it, like this way. So now it's both sides are back, so that I cannot cheat. Okay. Both sides are back, and I cannot cheat. And then I put it in front of me, and then I move it slowly to the side, and then stop it when I feel like I cannot see the color and identity of the card anymore. And then at that location, I continue to see my finger in front, and then I flip the card, and then I say it's probably like red 6, probably heart. And then the answer is, oh, so this is 6, but it was not red, and it was clover. But it's kind of that this is a kind of the experiment that you want to do by yourself. Okay, please do it right now if you can, if you have a trump card. right. So, okay, so this is the first experiment of the your phenomenology, and then here we have this randomness component that if you shuffle the card enough, then you can make it random, and then you can compute the number of the times that you're correct and the incorrect with your confidence rating and so on. So I was correct in terms of 6, but wrong in color and shape. And if you repeat this together with the confidence rating, we can, in principle, measure the objective performance and also metacognitive accuracy of our peripheral performance. Now, let's try it by yourself and then answer the Moodle question for metacognition on your peripheral vision. If you have observed something, then please also say that in the forum. Okay. And here is one schematic proposed by the Cohen, 2016, and there they are claiming that consciousness is sparser than we think. Okay. So here's the one schematic that was proposed by Cohen and so on in 2016, saying that consciousness is sparser than we think. And they claim that we still see far less than we think we do. And here's the demonstration. So where the card has to be actually be for participants to identify it. And then they say that where participants predict the card will be when they can identify it. But I'm not so sure about this. I'll see what you guys get. In my own experience, I tend to be more or less, in a sense, metacognitively accurate in that, I put my card over there so that I was able to actually identify number 6, right? And also, but the color and also shape-wise, it wasn't correct. But I didn't actually, force myself to put the card into the location where I can identify the card color or card shape and so on. So, we can do all these things empirically. This depiction to me feels a bit of the over-exaggeration, but we'll see what you feel. Okay. And then let's go to the next one, which is the peripheral experience of the color versus motion. Okay. And this relates to also what we learned this week. Here, again, you put your finger over there, and then you do the same thing, but then the relaxer condition, okay? Estimate whether you can see the color of the card. Okay, so basically you move the card until you feel like you can see the color. Okay. Then flip the card, and then locate the card where you cannot see the color of the card, and then move yourself, or better if you ask people to move it. Can you see its motion? That's the so the dissociation of the color versus motion in our visual field. So how it looks like. So you again put your card and then move it until you don't see the color of the things, and then here is more or less like I don't really see the color of the stimulus very well. But if I juggle it, or better, if somebody else does it, then what you will notice is that the point where you don't see the color of the stimulus very well, but you see the motion of the stimulus very clearly, and that to some people is going to be a bit surprising. You can see some property of the stimulus, but not others. And then another kind of the demonstration would be like ask another person to move the card from very periphery, and then say stop when you start to see the motion of the card. And then after that you can see whether you can identify the color and number of the card there. So this is basically the same thing. To dissociate your sensitivity to the motion versus color. And then after you try it out, and then you can also again, come to the forum or answer to the survey. Okay. So here's again the Cohen schematic. Stop. I see the motion over there. Say stop. You see the motion. And then what color is the moving object? If another person asks, then no idea. Red, blue, or wow, that's surprising. But I'm not so sure whether people are really bad at this type of experience. At least in my experience, many people spontaneously tend to say that they can locate and detect a motion in the periphery, or let's say like an insect, without knowing what it is. And then to know what it is, they make an eye movement or head motion to look at that. And so if that is correct, then basically people know that we know something is moving without what it is. And if this is the case, then their claim that people are people don't see things than they think they do. Okay, so that the dissociation between the metacognition and the color and the motion, this is posed as a sort of a demonstration here, but I'm not sure whether anybody has actually quantified it or empirically tested it in a large scale. So it's one of these issues that are open now as a consciousness science. You are now entering into the, as you see, this paper was published in 2016, and Cohen is the author of some of the papers that you presented in a journal club, and this is still like much debated kind of topic. We are consciousness researchers have been coming up with various different kind of measurements and the stimulus, and then also in this case the virtual reality or widescreen vision, which was not available before, and then to test these things in a more controlled manner with controlling different kind of situations such as expectation, instruction, and attention, which we will go into the detail next week. So this is one of the topics that you can also propose for the final project. We are, coming to the border of the knowledge. Okay. So just to reiterate, that to me it's quite, clear that the color perception in the periphery is real. But one of the most, influential philosopher called Daniel Dennett, said something like the following in the Mindscape interview hosted by Sean Carroll. Let's hear what he says.
If I try to abandon that intuition, I don't know what to trust.
Yeah. Well, it calls up the reliability of the usefulness of introspection generally, right? I mean, introspection is where we get a lot of these ideas of our experiences. Should we be generally skeptical of introspection? We learn something from it, certainly.
We learn something from it, but yes, we should be skeptical. First of all, let's start small and build up. I love to point out to my students and others all the ways in which their consciousness isn't the way they think it is. For instance, it seems that our color vision goes way out to the edge of our vision. It doesn't. It seems that we have high resolution vision out to the side. We don't. I love to point out that lots of things that are surprising, I can demonstrate to them. I think, whoa, who knew that? Well, you didn't, did you? So forget about the so-called incorrigibility of first-person acquaintance. That's just a mistake. That doesn't mean that we're not reliable informants to ourselves and others about many features. But forget about this Cartesian idea that on the inside, we are the masters of what's going on.
So you're saying that even when we experience the outside world, there's a lot of stitching in.
Okay, so that's probably enough for our purpose. But if you're interested in hearing the Daniel Dennett's talk and conversation with Sean Carroll, a physicist, I recommend you to go to subscribe their podcast or YouTube here. So what Dennett was saying is he's so-called illusionist by the category of consciousness, researcher within the philosophy. So he basically thinks that all the conscious experience is illusion, whatever illusion means. Illusion itself is experienced. I don't really understand what they want to say actually, but anyway, he pointed out that we should be skeptical about our introspection, which is totally fine. I'm also I have no problem with this. And then he goes on to say that we think the color extends to the peripheral vision, and he quickly dismisses it doesn't. And then we feel like the resolution of the experience is as high to as to the periphery, and we don't. But it isn't. That's what he said. I don't know really what he means by this. First of all, color is likely to be experienced, and it is experienced for many measures in the periphery, first thing. And another one is that we do not most likely consider the peripheral vision to be as accurate and as high resolution as a fovea. Most people try to look at what's there in closer look by moving eyes or heads. That means that they know that it's not really clearly seen over there. So he said something like this over and over again, and doesn't change his attitude over the 30 years or 40 years. So I don't know what to do, but this is again the reason of why I'm kind of bringing this issue to the front and then discuss about this. It is again one of the most controversial kind of things in philosophy or cognitive science. And for those people who know the vision science better, I think their attitude is much less radical than this. But let's see how it goes. Okay, so in summary, I just wanted to discuss, and this part is in a sense a bit more philosophical or phenomenological than other parts of the lecture today. Experience in the periphery is at least I would say controversial. And please answer the question for the metacognition on your peripheral vision in the Moodle. I'll see what you think. Okay. So the next video is about role of eye movements in conscious vision. Okay, so here is the lecture part 2 of the week 5, and then we are going to talk about the role of eye movements in conscious vision. The last part of the video might have missed some of the slides, so I'll just go quickly. So after this, we talked about motion and its metacognition of the peripheral vision, and then I also introduced the Dan Dennett's video and radio show here. So if you didn't see the slides together with the lecture contents, just go and slide the page to see what the link was. Okay, and then this is a summary, but I didn't have much slides to say. Okay, so roles of eye movements. So Dan Dennett and other people who claim about the illusory nature of this peripheral vision tend to make a reference to this figure. So here, as I mentioned in the last initial part of the lecture, important thing to realize is that these axis on the y axis is density, okay, and also it's not 0. It's always above 0 for the cones. So there is always a chance for the cone to be active to explain the color phenomenology, and how vivid it is, that's a question. But one of the things that all these people also say is that you actually do not see the color in the periphery, at least very well, because of the lower density of the cones at the periphery. But somehow we feel color in the periphery, okay? And the why, that's needs to be explained. So, One possible explanation is this. Also, because we move the eyes all around to see color in the periphery, and we are unaware that we are seeing the color at the fovea. So every time we feel like we see the color in the periphery, it is because of the memory and the expectation of the color that we directly look at the fovea, and then comes back to the other location, and they still remember or reconstruct the experience around the periphery. That's one explanation. Okay, here the eye movement comes to the center of the scene. One important fact to remember, which I might actually examine in the quiz, is that we move the eyes 3 times per second. But most of us are not really aware of this fact. 3 times per second is relatively a lot, but usually we are not aware of each of the eye movements. And this is a big puzzles of eye movements and the conscious vision, actually. And as I said, you move the eyes a lot, but are you normally aware of each eye movement? If I ask, most of the people probably say no. And then if I also ask, can you become aware of when your eye movements happen? Then if you pay attention to it, most people say yes. For example, if I say, like, oh yes, there was an eye movement, yeah, I can totally be aware of that if I pay attention to it. But normally, no. And this already hints at the relationship between the attention and the consciousness. And something that we will be discussing starting from next week is that whether attention is always necessary to become aware of the movements, such as eye movements, actions, and so on. Okay. And then the third question is that during the eye movements, can we actually see things? This is also, again, a very, very fundamental question. If there are 3 eye movements per second, and if we are not aware of it, what are we actually experiencing during this eye movement? This is really weird. Okay. And you normally do not think about this. But once you start to think about this, some of you may not be able to sleep at night. This is very weird kind of situation. So again, eye movements and conscious vision. To see this effect, what I want you to do is the following demo. Okay, face to your friend, face to face, or record your face with a video. Okay, like Zoom is one way of doing it. And then try yourself or ask the friend to move the eyes from right to left like this. Okay, maybe if I just stop sharing the screen here, and if you see it, my eyes like this, then the question is whether you can actually see my eye movements. Okay. Now, then the important thing to try is for you to go in front of the mirror and then look at your own face, and especially pay attention to your left eye, and then move your eyes or your focus to your right eye. And then see whether you can see the quick eye movements of yourself. Okay. If part 1 allows you to see other people's eye movements, then that means that your vision or conscious vision has enough sensitivity to see the quick eye movements by eye. Okay. However, if the part 2 says that by doing this part 2 demo by yourself and you can't see your eye movements, then there is something interesting going on. You are not seeing things while during your saccade eye movements, quick saccades. Okay. And in fact, this is known as a saccadic suppression in vision science. So here is the paper coming relatively recently, but the phenomenon itself is known for a really long time. It's called saccadic suppression. So what this type of the research does is, for example, ask the participants to look at some kind of target initially, and then make an eye movement, a saccade, to this, bright spot as fast as possible. Okay, and then during this period, they also present some kind of a patch here, that is either bright spot or high contrast or something like that, and then ask people to see whether there was something over there or not. It's a signal detection task, and then using a closely timed control measurement device, they can control the timing of this flash and also duration of the flash. And then what typically is observed is that depending on the stimulus, but the proportion of the correct trials, let's say, if there is a 1 in 4 chance of the success, 25%, people are really good at the detection of this thing, as long as if it's 50 milliseconds before the start of the movement of the eye. Okay, but even before the starting the movement of the eye, you start to not to see this kind of thing. It degrades quite a bit, and then just before 25 milliseconds or something like that, you can't really detect it reliably, 50% or 70%. And then during the saccade, you are making eye movements itself, and that can extend from 0 to 50 milliseconds or so, depending on the distance you travel. It can be longer distance, longer duration, and shorter distance, shorter duration. And then after eye stops moving, in this case, this particular paper doesn't align the response with respect to the offset of the stop of the saccade, but let's assume, which is probably a reasonable thing, that in 50 milliseconds after the saccade onset, that's the time when eye stop moving. Still, you can't really see and detect this kind of stimulus. And it takes roughly 100 milliseconds to recover the normal visual threshold. Okay, so what it means in our life is that every time when we make an eye movement, before and after, there is a period where you are not seeing things very well. And that happens 3 times a second. And still, we are not really aware of this period as a blank period or anything like that. So our experience is likely to be more continuous. However, depending on the time of the saccade, there is a significant editing going on, so that something that happens before and after is stitched together to make it continuous kind of experience. Okay, so let's move on. And eye movements, so that's a fact about saccadic suppression. And the next is that when we make an eye movement, how fast we can make an eye movements to the target. This becomes important to design the experiment where you want to exclude the possibility of contamination of the results due to the eye movements. But this is just one particular example of the experiment by Kirchner and so. But what it shows is that when you have a fixation cross like this in the beginning, and then that's presented for roughly 1 second to 1/2 second, and then it disappears. That's actually important thing. Disappears for 200 milliseconds, and this is a fixed time. And then if you flash the image really briefly, then people can make a saccade to the direction of the animal or face or target car or something like that. This is called ultra rapid vision. So this is called electrooculography, EOG, and then it measures the voltage and diffraction around the eye by putting some kind of electrode, and then you can see that there is some kind of fluctuation at the baseline, and then 0 time where the people presented the picture. Within 120 milliseconds, 125 milliseconds, I can start to depart to this target location here. So that's the location of this one. This x-axis position is converted into the voltage here. That means that the brain can process the image within 100 to 25 milliseconds and already execute eye movements. And this puts a bound, that lower bound of the recognition capability of the visual system to be roughly at least like 80 milliseconds or so. Otherwise, there is a time to take for the neuron, the brain to compute the eye movement signal, eye movement plan, and then execute that. Whether this is happening at the conscious level or not, that's unclear right now, and it's again active area of the research right now. We don't know the answer whether this is a consciously mediated or not. Most likely not. Okay. But for our purpose, what's important is that even at this extremely idealized situation of the eye movement, where you remove the fixation point, this actually is very important to reduce the inhibition. And then if you know the timing of the stimulus, then you can finally make an eye movement at 125 milliseconds. So that means that if you remove some of these conditions, then your eye movements can take roughly 200 milliseconds or 250 milliseconds. And further on, by combining this, our knowledge from the saccadic suppression, we can also estimate that the vision before and after the saccade is also not available to the brain. So this means that we can reduce the eye movement confound when we are probing the experience in the periphery. So what it means, the most direct way to do it, to examine the philosopher's claim, such as Dennett, we can measure the eye movements. And this is becoming easier and easier, and some of the virtual reality kind of goggle can have it inside. And so, this is another interesting direction for the consciousness research, but, measuring the exact eye movement, like, the paper I presented, it's going to be important, or present the stimulus for a brief period, like less than 250 milliseconds, and present them in a random timing and position. And then that way, we can remove the possibility that, any saccade eye movement towards the stimulus in the periphery makes the vision blurry and also less visible. And any incoming signal is strongly attenuated. And therefore, we can remove the confound of eye movements, expectation, and attention, and so on. All right, so I introduced Ariel's task last week. And if you haven't done that, I would encourage you to participate in this similarity task by following this link or link in the Moodle. And basically what we are asking you to do is to make a judgment about the similarity of the color in the fovea and also periphery in the display, and then to ask whether you can actually make a consistent and a very good judgment about the color at both of the locations when we adjust the stimulus size. Okay, and if you haven't tried that, I encourage you to do that. Okay, so then the summary, which is not conclusive at the moment because this is as I said, still controversial kind of topic. We fooled the experience color in the periphery because of the non-conscious eye movements, and most likely brief presentation of peripheral stimuli can address this question, and your own experience and your own judgment will matter. And then let's see whether that's going to be either similar or different at the fovea or periphery. Okay. And then as a side note, I would say that we cannot see well during the saccade. This is saccadic suppression. And as I said and emphasized, saccadic suppression should be happening every, in the beginning and the end of every 3 saccades per second, but we don't see it. And what we are experiencing is, in a sense, an edited word. And how does it, come about to this experience is really one of the hard problems that we are now getting, closer to. So the next part of the video is going to talk about this neural mechanism of the peripheral vision. Okay, so we are now going to cover what are the neuronal mechanisms that are responsible for peripheral vision. And here we are aim to explain the property of the visual system from eyes to V1 and explain the color vision at the periphery. So this is just a recap of the retinal structure going, the lights are going from top and then goes through the layers of the ganglion cells here, and then reaches in the end to cones and rods area, and then these layers are intermediate pre-processing areas, bipolar cells, macroin cells, and then reaches to ganglion cells, and then axons go like this way, and axons are this yellow thing on the figure. And then if you do the neurophysiological technique, then you can record the neuronal activity from this retinal structure. The important thing is to insert the electrode into the eyes, and here the eyes doesn't actually have to be inside of the live animals. Eyes can be isolated but still respond to the lights. So for example, glass electrode can be penetrated like this and then attached to the ganglion cells in the retina, and then you can record the spike that looks like this vertical line here. It's called a raster plot. And at the same time, you can also potentially measure the same axons that goes from here to here, and then measure the spikes at the optic nerve level here. And then basically this scheme shows that, looking at the same, as long as you're recording from the same neuron with the axons identified, then you see the same pattern of the spikes. And the spike is, as you can see, on and off, and then the pattern of the pulse and also temporal and spatial pattern of the pulse is conveying the information about this visual information. So, and what you're learning right now is this very important concept of the receptive field. Okay, and in particular, in the case of the retinal ganglion cells, receptive field is important. So each of the line corresponds to the particular response over 2.5 seconds of the visual stimulation. And then the top is when the particular retinal ganglion cells recorded are shown by the black thing under their so-called receptive field. So here it is, in a sense, the baseline firing rate of the retinal ganglion cells. And then when you present the spot of the light here in the left side of the neuron, and then this neuron actually spikes a lot during the stimulus presentation time here during this on to off period. Okay, and if you convert this spikes into the sound, then it sounds like that's a neurophysiological recording. And then if the optimum size of the stimulus as a white spot exceeds, and then if it stimulates too much, then it doesn't produce as much like kind of response. On the other hand, the optimum location, in the optimum location, if you present the black thing, on the other hand, then this becomes inhibited. It becomes below the baseline. So one would conclude that this particular neurons that we are recording from, has a receptive field that looks like the center area that becomes activated by the positive or sort of the bright spot, and then it's suppressed by the dark spot in the center. On the other hand, the annulus of this center area because it inhibits compared to this, central spot stimulation. This area is called the inhibitory area. So most likely this neurons receptive field looks like this. Center is activated by the white spot and inhibited by black, and the surround is suppressed by the white and then less suppressed by the black. So that's the receptive field of this particular neuron. And then similar thing can be applied onto this analysis of these neurons. Here, the baselines again identify the black spot, and then white spot onset inhibits this neuron. But interestingly, here releasing the white spot into the black makes firing here. Okay. And then white circle, bigger circle, doesn't produce much. It's similar to the spontaneous rate. And then now the black center white annulus pattern excites the neuron a lot at the onset, and then at the offset it becomes slightly inhibited. So two things about this neuron. So this neuron's receptive field is likely to be that center is excited by the black, and then also excited by the white ring here. But this white ring excitation is strongly inhibited by the white center, and then that's consistent with this one. And then also one thing I forgot to mention on this thing is that there are two types of the response in terms of time. Stimulus onset response, which is easy to understand in terms of mental kind of picture here. That's usually called onset response. Okay, and then at the offset of the stimulus here or here, that's called offset response. And typically, retinal ganglion cells have, in the end, have a structure called on-center or off-center. So on-center corresponds to the left and the off-center corresponds to the right, and also a combination of onset response or offset response. So at least there are 2 by 2 kind of response matrix, on center or off center, and then onset or offset response, and they are all found in this neuron. And this onset and offset response is pervasive across all the cortical areas, and we don't usually talk about offset response, but you can assume that many of the neurons have this onset and offset response types. Roughly, I'm not sure whether the exact proportion is computed, but maybe half and half could be assumed to some extent, because nothing counts as a background. So background is onset for something, and onset for some cells could be considered as a background. Let's move on. And then this is another important figure, and I'll explain. So this is the visualization of the retinal ganglion cells dendrites on the visualized on the surface of the retina. And the one type of cells called a midget cell, that means small, and the parcel cells that are bigger in terms of like a big parcel. And the midget cells are usually connected with the cones, and the parcel cells are connected with the rods. And the parcel cells are related to the detection of the motion or low contrast stimulus. As you can imagine, some kind of functionality that goes with a bigger receptive field. Basically, the size of the dendrite also corresponds to the extent of the receptive field. Okay, and the midget cells are small, and so it's correlated with sort of the fine resolution and also color type experience. But exact computational wiring is not known exactly, at least in the human. Okay, and then the important thing is that first, the size of the neurons are indicated by this scale. So here roughly it's a 115 micrometer diameter neuron. And this is much bigger, roughly like 250 micrometer neuron. And then the numbers below here, 1 millimeter, 5 millimeter, or 10 millimeter, these correspond to the distance from the fovea. Okay, so if you remember this, eyes, figure, fovea corresponds to somewhere here where the axons dips here, and then at the 1 millimeter from here, you find relatively small one for both midget type and the parcel type, and then 5 millimeter, you get bigger retinal ganglion cells, and also 1 centimeter, 10 millimeter, you find even bigger neurons. Neurons with bigger dendrites and also bigger receptive field. And this is very important for consideration of our discussion. Okay, so what it means in terms of our fovea peripheral vision. So as I said, the central retina, closer to the retina, each of the ganglion cells actually projects to almost one-to-one kind of no transformation is happening from retina to the next stage of the visual processing called the lateral geniculate nucleus. And I will spell it out in later. And then at the level of B1, peripheral primary visual cortex, these the LGN neurons tend to converge onto one neuron. So B1 neuron's receptive field is even bigger than the retina or LGN. And it also has a line-like shape, as I explain next. But importantly for our discussion of fovea versus periphery, the peripheral neurons are already bigger in terms of the receptive size at the level of the retina. And then it converges on the V1 neurons like this way. So V1, at the level of V1, the extent that it can collect the lights of a particular wavelengths, like yellow, can be actually bigger. So when I presented this type of the figure in the demonstration, last week and also at the beginning of this lecture, when red dot is very small but presented at the fovea, it can project to certain clusters of the retina and then excite a particular neuron in the V1. And if these guys prefer or generate color experience of the red, most likely it's not even like B1. After that, B2, B4, and so on. And then current theory says that B4 or B8 is likely to be the color generating center of the cortex. But by that time, when the signal gets there is some kind of convergence of the information, but still, all these areas of the neurons are looking at particular relatively small areas of that vision, and our perception is reflecting on that small part of the visual field. On the other hand, the big chunk of the yellow here projecting onto retina of this wide side, that can effectively stimulate these neurons, which prefers yellow light, let's say. And then that projects to LGN, and that converge onto one B1 neurons. And you can imagine that this thing actually goes on to B4, B8, and this generates yellow sensation. And there is nothing magical about that. So what it means is that as long as the stimulus is presented in a large extent or in proportion to the scaling of the magnification or the scaling of the receptive fields of retina or B1, then you can expect that fovea and periphery can have a similar property of the visual experience. Okay? And that's what you probably experience in this type of the visual stimuli. On the other hand, just to summarize the last figure, so neurons that respond to the peripheral visual inputs reflect inputs from cones of a wider area than those that respond to the foveal visual inputs. And also, this convergence effect becomes stronger for the neurons at more periphery. Assuming color qualia arises arise from higher areas, big and uniform surface can evoke stronger color chorea. Okay, so that's the upshot of the last figure. On the other hand, let's think about the another demonstration that I gave. Okay, so here the size of the stimulus is uniform across all these fields, and then in that case, red at the fovea can be perceived as red, like this mechanism. On the other hand, that the peripheral stimulation, like here, projected onto this peripheral patch path, this does not contain the uniform color stimulus, and especially it is interspersed with these gray areas, and it doesn't effectively generate a response from either pink or green or purple. And then as a result, it doesn't convey the vivid color kind of information. And further worse, these B1 neurons get convergence from other neighboring neurons that also receives an unclear kind of response. And in the end, it doesn't generate very vivid sensation at the color area in the higher area of the brain. Okay. If the same size of object is projected to the retina, then neurons at the fovea have a better spatial resolution, and collecting inconsistent color information at the periphery predicts that more blunt responses of the neurons are responsible for color chorea at the periphery. So there is nothing magical about that. But this is not the same thing as the statement like Dan Dennett made, as we cannot see color at the periphery, or color extends over the edge of the the vision. It's not really, you can't make that kind of claim from just by this type of the argument. Okay, so summary is that of this section is that retinal ganglion cells have a receptive field. That's a very important concept. Once again, I'll highlight that looks like annulus or on center, off center, and send visual inputs to the lateral geniculate nucleus through the through spikes, which is either all or none. And then some are sensitive to onset, and the others are sensitive to the offset of the visual stimuli. And the receptive fields of the retinal ganglion cells scale as eccentricity, and that has an implication for conscious phenomenology in fovea and periphery. So the next part talks about part 2 of this part. So what are the neuronal mechanisms that are responsible for peripheral vision? And now this is going to talk about the more focusing on the property in V1. So this is the figure 4.1, chapter 4 of the Christoph Koch's book, which depicts different types of the neuronal cell types within V1. And here you will note several things. One is that the type of the neurons, really many, and one of the most important or well studied neuron is called pyramidal cells, and that has a shape like this, triangular pyramid-like structure of the cell body that typically extends the dendrite, upward, and then that branches a little bit, and then projects the axon to the bottom. Okay, and then that goes into other areas if it locates into the layer 5 and so on. And then many of the neurons are much smaller, and then some of them are smaller neurons tend to be inhibitory, and some of them are excitatory. We can't tell from the shape of that. By the way, these pyramidal neurons tend to be almost exclusively excitatory, meaning that the neurons that receives the output from these neurons are becoming more excited and generate the spikes. Okay. And then the second component is the mysterious numbers here. And this, and also you can kind of see the band structure here for each of the areas actually. And each of the cortex tend to have this 6-layer structure, 1, 2, 3, 4, and then 5, and then 6. And if you ask physiologists, sometimes they say that this layer band structure is visible just by the sort of the microscope, and sometimes you need to do the staining. But important thing is that each layer of the within each layer, neurons tend to have a similar connectivity patterns with respect to other layers of the neurons or other cortical areas, and that's that leads to the notion of the hierarchy of the visual cortex, as I explain next. So this is an very important concept in vision science called the cortical hierarchy. And it's proposed by this Feldman and Van Nessen in roughly like 30 years ago. So what they these anatomical researchers found was that if the layer 4, which is usually considered as a middle layer, and then layer 2 and 3, that is upper layer, and then 5 and 6 is considered as a lower layer. And why we don't have layer 1 is that the layer 1 tend to have only axons or dendrites and not the neurons, actually neuron cell bodies. But anyway, the location of the cell bodies, either in the upper or middle or lower, as well as that axon terminals location in the layers, can be considered into roughly 3 types. One type is a forward or ascending, another one is a lateral, and another one is a descending or feedback. So by feed forward, what it means is that some kind of the area that projects to, let's say, area V1. In the case of the LGN, we don't have a laminar cortex, but the areas input into cortical area tend to terminate in this mid layer, but not in the upper or lower. And that is true for LGN to V1 case, okay? And V1 to V2 case, that's also like this, and so on. So that's called a forward or ascending type connection. On the other hand, descending type connection, such as V2 to V1, or other types of the feedback connection, which is going opposite of this ascending direction, that tends to originate from upper or lower layers, and then terminate in the upper or lower layer. It could be coming from only lower layer and then terminate in both. That's a typical descending projection. And the lateral corresponds to the similar level of the hierarchy, such as the most prominent is probably MT to B4 or something like that. I'm not an anatomist, so I may be wrong about this, but these tend to have upper or lower areas of the origin, and then projects across all the layers. And then based on these types of the terminal projections, what Feldman and Van Nessen did is to generate some kind of maps. So that corresponds to this, sometimes called a Feldman-Van Nessen diagram. It looks like on the train's, line or very complicated metropolitan road map, but it is according to this, anatomical hierarchical assignment rule. It is constructed. And here you know that retina initially goes to LGN, and by the way, hierarchy is assigned from bottom to the top in this way, and then LGN projects to B1 in this way, and then B2 is located above B1, and then from there many different kind of areas exist. And what we talk mostly in the lecture is like B4 and MT. MT is for the visual motion, and B4 is for color and object recognition. And also the clusters of inferotemporal cortex called IT, as well as the intraparietal areas, and so 7A, 7B, this is called parietal areas. And then here is a hippocampus that is famous for memory, okay? And this type of the hierarchy can be assigned by having this anatomical connective pattern across the layers. And for now, we just stop here, and we will come back to this type of issue later. Okay, so then the another important structure of the cortex is that V1 neurons have a different kind of a tuning. Okay, so unlike receptive field for retinal ganglion cells, this neuron, for example, let's say, if you are fixating at here, then the neurons might respond to the stimulus that is presented somewhere here, bottom right, okay? And then it's not only the location, but also the type of the stimulus that matters. And it took really long time for the neurophysiologists to realize this, and that led to the Nobel laureate discovery of the Huber and Wiesel. And you might have heard of this type of story, so I'll omit it. But what they discovered is that not only showing some of the stimulus in this receptive field location, but you need to have a proper stimulus. And in the case of the V1, that proper means many different things, but for example, it's the direction of the motion and also line orientation. Here, this particular neuron responds really strongly and onset response with this angle of the line, and it's responding only to this direction, but not in this direction. Okay, and then if you change the orientation, then you find that this direction is really optimum for this neuron, and that's called the direction selective cell in the V1. Okay, so the higher the hierarchy a neuron locates in the area of the brain, it tends to have a bigger size of the receptive field, and that's because of this convergence property of the neurons. So any given neuron tend to have an input from thousands of the neurons, and that represents each different spatial location. So the V1 neurons tend to have a bigger receptive field compared to LGN or retinal ganglion cells, and B2 has a slightly bigger, and B4 even bigger, and then IT it's much bigger, and we'll come to that. And that's related to the concept of attention, as you will see. The second is that the higher the neuron locates in the hierarchy, the property of the response that optimize neurons firing becomes more and more complicated. And here you will see the direction and the orientation of the line that was not present in the receptive field structure of the LGN or retinal ganglion cells. And then if you go to V2, it adds something, and go to MT, it adds something. And the bulk of the neurophysiology has been spending on spent to discover this complication of the receptive field across many areas, and that's still going on. Okay. Then the third important neuronal structure of the V1 is that what we found, what I did, already mentioned was this vertical direction of the layer structure, but here horizontally, the neurons' receptive field properties become similar across the horizontal direction. Okay, and that's called the columnar structure in the cortex. And in the V1, there is two, at least prominent columnar structure that can be visualized by many different kind of experiment. The one is ocular dominance, and that becomes important for the next week binocular rivalry lecture. So what it means is that at the layer 4, for example, within V1, neurons within this left or right columns of the columns are locating this area, they tend to respond only input from the left eye, but not to the right eye. And then here, neurons respond to only right eye, but not to the left eye. Okay, that's the ocular dominance column. And further, within the ocular dominance column, there is an orientation column. So here, the neurons respond only to the horizontal line. Here, neurons respond only to the vertical line. just to make it clear. So this one is horizontal and this one is the vertical. And some of them only responds to, let's say, this direction of the motion. But, sometimes this is even split, and then this half responds more to this direction, this portion responds to the other side. It's a direction dominance column as well. That's also discovered in the V1. Okay. And here is the just peeking of for the future lectures, but orientation column structure for the MT. This is MT. On the left side is MT. Is both motion direction as well as the depth of the plane. If the motion is happening near you or farther away, that also has a tuning, or a property of the receptive field is dependent on this kind of properties. And this one, that looks a very complicated column, that's called inferior temporal cortex. And they have a cluster of the neurons located within this area that responds to monkey face or some kind of shades of the circle or cube, the sphere, and then elongated, circular shape and so on. So that's found in IT. Okay, so that's the important feature of the V1: layer structure and the more complex receptive field type, and then columnar structure. And then another important property of transformation from LGM to V1 concerns how does the visual image map onto V1. Okay, this becomes very important when we consider the phenomenology and the property of the V1 here. And in an upshot, I will introduce this concept of the retinotopy and the cortical magnification. Okay, how does it work? So if you are focusing on the right visual field, let's say you are looking at the center here and then don't move your eyes, then let's imagine that you are looking at with your right eye, and then there is a blind spot here, roughly 10 to 15 degrees of the eccentricity, and then there you don't have any retinal ganglion cells for the right eye that responds to this part of the brain, the part of the visual field. Okay, visual field refers to the outside of the world, and then this gray area is the one that you can see only with your right eye. And then the white side is the one that you can also see with your left eye. So you can check by closing your right eye or left eye, your visual field actually changes. The most, farther away is only accessible with the right eye for the right visual field. Okay. And then now, what happens when the image goes transformed into the V1? So this is the final figure. Here there are several important things happening. So in V1, the world is not projected like this half circle. First of all, that's a very important thing. It looks like elongated ellipse kind of shape. And if you know the mathematics, this is called a log, complex log mapping. What it means is that it emphasizes the representation of fovea. Let's say this is a 2.5 degrees, so it only corresponds to this much of the fovea of the right side, projects it into the, also by the way, this goes onto the other side of the cortex. Okay, left primary visual cortex. And this gets mapped to this large spot. On the other hand, this 40 degree and everything else gets projected even smaller than central 2.5 degree visual degree angle. And then blind spot gets projected here, and then this part is dominated by the input from the left eye, actually. And then you'll see that, roughly 10 degrees of the visual angle, which is roughly speaking half of the display size on your, laptop with arm's distance. That already explains 50% of the V1. Corresponds to 10-degree visual angle. Okay, that's very important facts about the V1 mapping. If I take stock of everything that I have told you, from retina to LGN to primary visual cortex, here is the important figure, okay? And I'll ask it in the quiz. Assume that this person is looking at the center of the display like this. And then the right visual field gets projected on the left side of each of the eye. And then the left visual field gets projected on the right side of the eye. So this is just, same as, the principle of a camera. All the image gets, inverted through the lens here. And then after the crossing of the optic chiasma, it turns out that, all the things that you see on the right side of your fixation gets to left LGN and projects to left V1. On the other hand, all the things that you see on the left visual field gets to map onto the right LGN and then right V1. This is very important, okay? And you can already imagine that, many of the folk psychological or folk neuroscientific notion of right brain doing calculation and the left brain doing arts doesn't make sense even from this perspective. But I will go into the detail of that in the later lecture. Okay, so now if you consider this fact, then how does the figure like this projects to the brain? That's a question. Let's assume that we are looking at nose of this person, okay? And then it should be the case that right side of this figure, including this left hand of the person, projects onto left visual V1, and then the left hemifield, left visual field should project onto the right V1. Okay, the reason why I cover this part with a question mark is because this figure has a little bit of the mistake, and it's actually really common that many neuroscientists and psychologists don't get this. So be careful about this on the website, based information. Okay. So just to recap again, let's go step by step. The V1 right visual field corresponds goes to the left visual cortex, and it's upside down, and the left right is flipped. Okay. So if we start with this, image outside, at the retina, it's all flipped, upside down, and the left and right. And then the left side of the V1 projects or processes this side of the thing. Okay, and that's also enhanced or sort of the magnified at the small part of the retina like this. It's gross, but that's how your visual cortex V1 represents or responds in this response, the outside stimulus. And then also, just to recap, or to make sure what you are understanding in more fully, this kind of Simpson picture, when you are looking at this Homer's eyes here with your fixation, then the left and the right B1 should project the image like this. First of all, what you see at the fovea the Homer gets represented like this, and then everything on the top right gets projected into the bottom left of the V1, and then other things you can check. So it's all flipped back and top and bottom and the left and right. Okay, so what are the implications for the conscious vision? So one question you might ask is that why do we not see the image upside down, like our visual cortex is projecting? It sounds silly question, but it's not, and also in the end it will lead to the hard problem actually. Okay, secondary, you might ask, why do we not see the image in a distorted way, like Simpson's projection onto the V1? What we experience is not the mirror image, and also in a sense, not the flip and flip up and down, and also left and right, but the actual reflection of the world in itself. This is not really straightforward, and in fact, if you have never learned anything about the brain, this might come as a potentially surprising thing. In fact, Aristotle is said to be hypothesizing that everything we see should have some kind of correspondence, in his terms, isomorphic correspondence in our head. But of course, we now know that red circular thing outside does not have the circular red thing in the brain. So it's clearly not the case. But why is not the case? It's not really simple kind of question. I mean, if you are asked by the kid, how do you respond it? I can't really offer a really good answer actually. And as I said, fovea is hugely enlarged in V1, but so that means also are we actually not seeing much of the periphery? That's also another thing that you might actually infer from this type of the data. And in fact, this is a really good conjecture. Although we do seem to have at least a space outside, and I don't think we see very well. And that fact that we don't see very well corresponds to the shrunk representation in V1. But why the space itself seems uniform, whereas the representation gets crowded or sparse, that's not super clear, really. Okay. So to summarize, this part of the lecture said that cortex is vertically layered and horizontally displaying the columnar organization. And at the higher hierarchy, the receptive field becomes bigger in size and more complex in its property. And the cortical magnification and the retinotopy, which is the correspondence between the outside world to the brain, represents the first step to the hard problem. What is the link between the neurons and phenomenology? Okay. And then another part of the summary is that the cortical representation of the fovea is huge in V1. Up to 2.5 degree visual angle corresponds to 20 to 25% of V1. And the good number to remember is 10 degree visual angle corresponds to the 50% of V1. And what does this mean about our impression of consciousness in the periphery? And then all of this summary, in a sense, I assumed or sort of implied that V1 is related to our conscious vision. But is it really the case? That's the question that we are going to address in the next week. Okay, but we have several things to do for the rest of the week, this week's lecture, so that's the next one. Okay, these are the important topics which will be coming back again in the next lectures. All right, so the final part of this lecture, this week's lecture, is going to be about the neuronal mechanisms that are potentially responsible for blindsight. So the key facts about blindsight to explain is first, their lack of visual phenomenology, at least their report. And the second, presence of the certain behaviors despite these reports. One is the eye movement to an object, and also localization of an object. And these are much higher than chance performance. And from this, Van Nessen diagram, I already mentioned about this, diagram. But if this is really all there is in the, visual cortex, then it's super weird that, retina to all these areas are actually, in a sense, blocked by B1 lesions. So if the B1 is damaged, according to this scheme, everything else goes, completely, defunct, and you'll get all the visual function to be completely nonsense, right? However, that's not the case in the blindsight. That's things to be explained. And this is a figure from Cox's books, the front page or back page of this, book. And if you look at carefully, this, the left, bottom left part of the retina. And by the way, this is a flattened representation of monkey brain. So this is the eye, and here is a lateral view, and this is a medial view. By now you know about this, lateral, and then this is medial. And then without disconnecting the connections, this figure generates this representation. And here, what you see is that retina on this side, the right side of the retina is projected onto this side of the LGN and then to B1. But there is this branch, superior colliculus and the palvinar, to go to V2. So this is an alternative pathway that is most likely responsible for blindsight. Just to get a better, view. So here's the magnification. So in terms of the number of the axonal connections, retinal ganglion cell projects to roughly 90%. Of the retinal ganglion cells projects to lateral geniculate nucleus in primates, but it's different for mouse. I think it's roughly 10% or so. And then the rest of the 10% goes to, or believed to go to, superior colliculus and palvinar. And that's the culprit of the blindsight. And here's the schematic that is usually invoked to explain the alternative pathway from retina to the cortex. And the retina branches to LGN, that is 90%, and then goes to serial processing from B1, B2, B4, TO, which is bulk of IT, inferior temporal cortex, and then reaches the amygdala. And the amygdala is a location that is responsible for many different kind of fear-related processing. And then retina also branches to superior colliculus, that's a 10%, and then goes to the palvinar, and the palvinar projects to the amygdala. And you see this magnification here. Palvinar is a part of the thalamus, and also lateral geniculate nucleus, that's also part of the thalamus. So LGN and the palvinar nucleus is actually, palvinar is close each other, but it's not directly connected. And then superior colliculus, that is slightly dislocated from LGN, that's connecting to palvinar. And then many of the literature on amygdala, which I also contributed, concerns the question of whether if the emotion of fear requires processing of this lengthy, serial processing, it may not be optimum. So some people thought that there may be some kind of shortcut from superior colliculus, palvinar to amygdala. And in fact, blindsight people tend to show some kind of a residual emotion processing, as we might talk about in the future lecture. But for our purpose, the important thing is that the superior colliculus is not only important for the subcortical pathway or the branch of the things, but it's also important for eye movements and attention. Okay, And then amygdala is important for fear or fear reaction and the fear experience. And that's a really simplistic view. And in fact, what we know from the anatomical research is that at least there are 10 pathways. And you don't need to remember everything. But what I discussed above there is this major dorsal lateral geniculate nucleus pathway that goes to the 90% of the things. And that projects to B1. And then that projects to B2, B3, B5, or MT, and so on. And then even within normal people, 10% goes to superior colliculus, to palvinar, and so on. Palvinar and superior colliculus. However, the Koei did anatomical study of the blindsight monkeys and then compared the projection. And if the B1 is lesion, after the long-term, reorganization, it turns out that, pathway to LGN even becomes, kind of thinner, And then superior colliculus and palvinar remains strong, and the perigeniculate nucleus, I'm not really sure what it's doing. It becomes bigger, and then other nucleus and also other stuff like olivary nucleus of the optic tract, medial or lateral dorsal terminal accessory optic nuclei, suprachiasmic nucleus, all of them have a slight bit of the branch, but I don't know much about what they are doing. This one, suprachiasmatic nucleus is important for the regulation of the sleep cycle or sleep-wake cycle. So we know a little bit about this, but not much about others. I mean, at least I don't know. Again, you are already coming to the front of the knowledge. Okay, so not only these, 10 pathways from retina to cortex, or at least, retina to brain structures, within even the brain, there are lots of shortcuts and also multiple pathways exist. So, for example, retina to superior colliculus to palvinar, the palvinar also has a major division of this part and also this part. And the amygdala seems to receive a strong connection from the part that does not receive the input from superior colliculus directly. But importantly, LGN also has an input to MT directly. So even B1 is damaged, retina to superior colliculus palvinar to B2 or B4, that's possible, as well as retina to LGN to MT is also possible. And then MT also projects to areas that are important for attention for parietal cortex or frontal and so on. And so these pathways are not really straightforward, and there are multiple pathways exist, and it also can change after the brain lesions due to the long-term plasticity. And this has an important implication even for our discussion on consciousness. For example, in the case of the masking, when the stimulus is presented for, let's say, just very briefly, one frame, and then followed by a mask for 40 milliseconds. And that means that initially, the eyes receive the input and the LGN, and then goes to the B1. So this part is a B1 in the case of the monkey, in a 40 to 80 milliseconds. And then next part that is actually activated is, in fact, prefrontal cortex called FEF. That is important in directing attention and also moving the eyes. And remember that superior colliculus has a potentially shortcut to there, and that may explain why this has an extremely short latency. And also, by the time it gets to feed-forward activation goes into the IT cortex for the face recognition and so on, then mask stimulus already reaches to B1. And so one relatively popular hypothesis of this explanation of the backward masking, why backward mask is so effective in erasing conscious experience, is that the time of input coming back from the higher area is roughly matching with this masking timing. So the initial response prepared sort of, the template of the visual representation and then feed backs into the B1. But when the time B1 receives the input, it is already overwritten by the mask. And that's why we don't see the stimulus in the case of backward masking. And that's one popular explanation of the backward masking. But for our purpose, we also want to make a reference to more complicated, reality of the response latency. 40, 50, 60, 80, 155. The many areas start to become involved, and also all the feed forward and the feedback inputs and the activation happens all over the brain. So reality is not as super simple like this way. Okay, that's what I wanted to make here. And then briefly, I also mentioned that spatial and also temporal hierarchy of the brain is important thing to consider, but it's also not really very well reconciled in the literature. So this is another sort of the frontiers of the knowledge in the brain science. And we will go into the detail for the next probably 2 weeks from now, that there isn't a hypothesis that says that from B1 of the monkey, it goes roughly into two pathways of the brain. One is for this dorsal pathway, this side is a dorsal and then this side is a ventral. And roughly speaking, people think that the vision for action or where and how to manipulate the stimulus and so on, these are all processed in this dorsal pathway. And this is hypothesized to be still remaining active for the blind side. So blind side has some kind of, shortcuts, let's say from stereo colliculus to MT, and then goes to this parietal dorsal pathway to enable eye movements and localization. And the activation of these areas are supposedly non-conscious. And that's one explanation of the blind side, why they can do the eye movements and the localization. On the other hand, why do we lose the conscious vision? Is because the conscious vision is mainly generated by this dorsal pathway. What pathway? And I'll go into the detail later. And this is a schematic of this ventral and dorsal pathway. Retinal, LGN, V1, and then this is already emitting stereo colliculus, but going to MT, which receives direct pathway from LGN and stereo colliculus, and then that goes to these parietal area that corresponds to dorsal non-conscious pathway, whereas these are the ventral conscious vision pathway. That's one schematic. Okay, in summary, the last part of this lecture talked about the blind side occurs when the retinal LGN B1 pathway is damaged, and the retinal stereo-colliculus, palvinar cortex, and the retinal LGN MT pathways are likely candidates for the blind side behaviors. And then eventually these behaviors are mediated by the parietal dorsal pathways. So the question is that if stereo-colliculus is actually important for attention, then can blind side patients still demonstrate normal attention to what they claim invisible objects? That sounds a weird question, but we are going to cover this in the next week. And then another thing important to summarize is that multiple pathways in visual pathway exist. And this complicates the temporal hierarchy and also interpretation of the Feldman-Van diagram. It's not the case that the last part of the field, the hierarchy, hippocampus, is expected to be activated at the last. In the case of the hippocampus, it is indeed slower in terms of response latency, But there are lots of shortcuts and also, alternative pathway, so that, Van diagram doesn't really translate into the expected latency of the visual response. And also, what is the role of the feed-forward and feedback neural activity for the conscious vision is something that we will extensively discuss. See you next week. Bye.